we focused on all of the main body parts of our plant, but we haven't spoken yet to flowers and fruits, which I think is of crucial importance when thinking about conservation into the future, because that's sort of what we utilize when we try to think about replanting systems or we think about the next generation of life in an ecosystem that we're trying to restore. So, in thinking about life on planet Earth, we have to, I think, go back to the origins of why we might have flowers and fruits in the beginning. And that also speaks to some of the other traits that we've identified, things like stomata, things like woody structures, things like roots, and essentially plants that exist on dry land. They are limited in the resources that are available to them. They have to find ways to function in a essentially dry world. So that's something that you don't see with things that live in aquatic systems. So if we look at things like algae, they're not going to have stomata. They're not going to have conduits to shunt water or other resources around because they're essentially immersed within the mediums in which they need to grow and thrive. And we see an evolution towards things like pine trees and things like sunflowers, what we call our gymnosperms and angiosperms, in the form of some of the early adaptations in vasculature and reproductive strategies in things like bryophytes. So those are our mosses, liverworts, and also pteridophytes, which are our ferns. I'm not going to go into detail on bryophytes and pteridophytes because I know there are some really excellent lectures that cover that offered already through Jepson that you can find online. But I want to really focus on gymnosperms and angiosperms because those are species that or those are groupings that we find prevalent throughout the California landscape. And they also speak to plants that are true seed producers. And the production of those seeds and the ways in which seeds and flowers are produced really speak to a lot of these constraints that I've already discussed that are imposed when we move to dry land. In particular, the need to protect that offspring against disturbance factors, the need to protect against desiccation. So let's start by thinking about gymnosperms. And gymnosperms are essentially all of our conifer species in California. That's basically just our conifer trees. But if you go to other places like South Africa, there are things like Welwitschia, or in other places around the world, you may have other strange things that exist within this lineage. But the defining feature of the gymnosperm is the production of what we call a cone. Gymno means naked, sperme means seed. And so what we see with a pine cone, such as this pine cone, which comes from Monterey pine, is that seeds will develop on these scales, which are these individual subunits that you see kind of wrapped one on top of each other like Russian dolls. And then at a certain point, those scales will open up. And as they open up, the seed is available just to fall out of the cone. So if we took this cone when it was in its open stage, we flipped it upside down and gave it a good shake, the seeds would just fall right out onto the soil. So they are, in other words, unclothed. They're naked and bare for the environment. And of course, this is also where pine nuts come from. And even while they're bare, they are coated in this thing called an integument, which is a really hard structure to break into. So I don't know if that's why pine nuts cost so much, but I have definitely had a hard time getting pine nuts out of their integuments. So something to note, though all of our California pine nuts are edible. In contrast, we can think about things called angiosperms, which are true flowering plants that also produce fruits. And so we talked about a pine tree. Here's a pineapple. The name and shape of a pineapple are kind of similar to a pine cone, and yet you could shake this thing all day, and the seeds that form within the pineapple, they are not going to be falling out of the pineapple. That's because they're enveloped in an ovary. That ovary is what we call the fruit. And so that fruit offers extra levels of protection for the maturing seed. So within our growth patterns and flowering patterns, we can divide things into a couple of categories. One is dioecious plants. Dioecious plants are those that have an individual that is only bearing male reproductive tissue or only bearing female reproductive tissue. The way we describe maleness and femaleness is that when we talk about female or egg bearing, we're talking about the larger sex cell, whereas the sperm, so something in the case of flowering plants would be encapsulated at first within pollen. That's something that is the smaller of the sex cells. So sperm is a smaller sex cell, egg is that larger sex cell. And with something like a kiwi, you find that there are distinct male and distinct female, so distinct sperm and egg generating plants. we find this in some cases here in California with things like Baccharis, which has that as a male and female plant. Then there's monoecious plants. And with monoecious plants, in this example of Douglas fir, we have an organism that is producing both male and female reproductive structures on the same plant. In this case, we're looking at the female cone here, which is that larger structure. And then you can see on the tips of the branches, the male reproductive cones that are producing the pollen that will ultimately land on and provide genetic material for reproduction in that female cone site. And again, you can kind of see just here in a very rule of thumb sort of way, those smaller cones are the male reproductive structure, right, housing that smaller sex cell, the female cone being larger, and also where the egg will develop, therefore needing more protection in the form of those scales. There's also a category called perfect monoecious, and that is something that we see really in our angiosperms, such as this flower here, Lilium colloquii. It's one of my favorite lilies. And it's sort of the textbook example, I think, in terms of form when we think about perfect monoecious flowers. That being that you have both male and female reproductive structures encapsulated within a single tissue. And in this case, we also have the addition of petals, which envelop that tissue. But I want us to kind of think about how we would get to something like this, how something like a flower that's so beautiful that you might see being sold, at your supermarket or floral stand could even emerge. And that was actually something that was really problematic for botanists and evolutionary biologists for a long time, because we see such diversity of flowers that exist in our systems. These are some wildflowers that exist just in the habitats that I study in the coastal grassland systems. There's such a diversity. They certainly serve some function, and yet the range of colors and patterns that we find are so distinct and unique. In fact, Darwin labeled this an abominable mystery because he just couldn't wrap his head around how these may have emerged. And yet we can look to flowers and see that they offer a lot of benefit in terms of protection, in terms of expediting reproduction, and also in terms of improving efficacy of reproduction and success. And again, these are flowers that just grow in the coastal prairies that I study. I didn't take all these flower pictures. I always have credited at the end pictures from folks who took these, but it's such an incredible array of diversity that we find here in California, and even just in a small patch of land. And just some more flowers, just because I like to share these things. But notice these different forms and functions, and we can also see here pollinators. So pollination will be an important driver, as we'll discuss, for this reproductive success. But in thinking about flowers and thinking about angiosperms in particular, these are also flowers, right? So we can look to Stipa pulchra, a grass. It's got those male and female reproductive structures wrapped into one unit. It is perfectly monoecious, and yet it lacks those petals. Or we can look at this alder and see that it lacks petals. It also has the reproductive structures separated, again, male and female structures separated out as being just, and yet even still, when we look at something like this, it doesn't really look very much like what we think of when we think of a flower. And undergrads that I teach often will mistake these for pine cones, because they do have a similar resemblance to that. And so I think that looking at these structures gives us a starting ground to begin to think and conceptualize what the origins of flowers may have looked like. And again, no one was there 130 million years ago when flowers first came onto the scene, but we can look at some of the structures that exist today as a proving ground for what those early developments may have looked like. So the first thing we want to consider, of course, is protection, and in having some sort of aggregation of tissue around our reproductive structures, we are affording some sort of protection. And it's not that petals, in fact, genetics tells us that these petals would have emerged from what would have at first been whorls of leaves, and that leaf tissue was essentially modified through some mutation over time to affect these sorts of more delicate flower structures that we see, these petals that are protecting the body and reproductive structures. But we also have to think about color. So people who are not from California often like to deride us for not having fall color and foliage displays. But if you go to the Eastern Sierras or creek channels, you'll see incredible displays from populace like Fremont's cottonwood, or in the case of the Eastern Sierras, Populus tremuloides. When we look at color in leaves, the fact that we see that fall change of colors it speaks to the first stages in deconstruction of that leaf. So as the leaf reaches the end of its lifespan and is no longer of value to the tree because there's not enough light for that leaf to be functionally viable, all of the complex structures that can be disintegrated are disintegrated and brought back into the leaf tissue. So that includes things like the nitrogen that we talked about before and the phosphorus and the potassium that are these mobile nutrients. We deconstruct those and take them back into the body of the tree. And what we're left with is all the stuff that we can't break down. So it's sort of like when you move house and you take all of your expensive possessions, but of course you leave behind the structure of the house. You leave behind the electrical unless you're particularly savvy and know how to remove that from walls, and the plumbing and stuff like that, right? That all stays. So we're left with carotenoids and light colored, colorful tissue that remains at the end of our leaf's lifespan. And so if you look at something like a bougainvillea, which has these flowers that are encircled with these really bright colored tissues, we're sort of seeing that same trend, but in reverse. And in fact, that gives us an insight into what would have led to some of those early colorations in our foliage, in our petals. And you can see that even in some native species like Vaccinium ovatum, where here we're first building that young tissue. It's bright red. We haven't yet built the photosynthetic machinery that's the most expensive, that might also be something that an herbivore might want to munch on. Instead, we're putting the less expensive chemicals in first, and also chemicals that may be a deterrent for something that would want to eat them. So that redder tissue, probably not as tasty as that greener tissue, which will also have more nutritional value to an herbivore. So that gives a little bit of background on color in our petals. Something else, though, is petals will obviously act to attract pollinators and also deter pollinators. And one of my favorite examples of that is Lupinus bicolor. And so that term bicolor speaks to how at the first part of the growth stage, we have these petals, what we call the banners, that upward portion of the petal, which is blue and white, right? And then after it's pollinated, that white area turns this lovely purple color. And what's happening is chemicals, after it's been pollinated, change the color of the tissue. And to the eyes of the bee, that pollinated flower suddenly has a no vacancy sign. Suddenly it doesn't appear as attractive. And so the bee will just avoid those already pollinated flowers and go to flowers that are still white. And so this allows us to start to think about some of the traits that flowers are using to attract pollinators. And of course, we spoke earlier about visible spectrum, what we can see. So we might see a beautiful yellow, I guess this is maybe a potentilla flower. We can also imagine that what the bee is seeing is something that's very different, or the pollinator is seeing is very different. In this case, almost a bullseye pattern that emerges on that tissue that would guide the bee towards the site of pollen in order to expedite that process and ensure efficacy in pollination. And other attributes we see as well, people often talk of how we may have co-adaptation or co-evolution between something like a hummingbird and a flower, so we get these longer structures. And then I think that raises the next question, which is, well, why would you want to make the nectar out of reach for your pollinator that has either that long beak or that has a long proboscis to reach into the flower? And the answer here is, again, we're trying to, as a flower, get as much of that pollen as we can on the pollinator. So you can think of, if you have a chip bag, when you reach your hand in for that last chip in the bag, right? That's when you get all that gunk on the chip bag coating your arm, right? And essentially, when we make the pollinator reach deep into the flower to reach that nectar, it's coating its whole face with pollen. And as a result, more likely to move pollen from one flower to another. Down at the bottom is an example of a flower that grows close to the ground. That's our asarum again. And the asarum has adapted itself to exist with ground-dwelling critters such as beetles and ants that will also pollinate that. So we see adaptation where there's mutations that emerge and are selected upon to match between pollinator and also the organism that is being pollinated. Something else I want to mention is shape of our flower. And this was brought up earlier as a question about maximizing or minimizing access to sunlight. Notice that the shape of our morning glory is sort of like the shape of a collector disc. So we thought earlier of how plants are accessing or redirecting sunlight through the shape and angle of their leaves. Here we have almost a satellite dish shape to our morning glory, which allows it to receive more incoming thermal radiation. And in so doing, and culminating that thermal radiation at the base of the flower, like a collector dish, what we're doing is we're speeding up rates of reaction, because when heat is applied, in the right dose, enzymatic processes are driven faster. And for a flower, you can imagine that looking at this tissue, that tissue, it's not green, right? It's not photosynthetic. So whenever you undergo sexual reproduction as a plant and you're producing things that are essentially a suck as opposed to something that is producing energy, that is going to be something that will be costly to the body of the plant. So you want to be able to drive these processes as efficiently and effectively and quickly as possible. And so being able to culminate energy, being able to minimize the number of enzymes you're using in that process have a lot of benefit when it comes to speeding up that process of reproduction and also minimizing risk in speeding up that process that something will damage that flower and make essentially abort whatever process of germination or of fertilization is occurring at that flower site. Of course, not all flowers will be pointing upward, and back to our lily, you can see that here we have a plant that is modeled in its shape, not unlike an umbrella. And you will see that flowers cultivate things like drip tips or have downward facing heads, essentially to avoid precipitation or other elements from damaging that tissue. So again, protection is really fundamental in the design of these flowers. And of course, we have to always realize that, I'm describing these as being traits that meet some sort of requirement, but we have to realize that the different ways in which plants are meeting these requirements are always going to be a product of some of those genetic underpinnings, the construction that exists that they can only manipulate uh, within a certain extent, and also that these processes are not consciously driven, but rather driven by natural selection that operates on whatever traits present themselves through mutations that become most favorable to the system in which that plant is growing. So I'll get some questions sometimes about seed collection as well and dispersal. And I think what we have to think about here is just as with flower diversity, the ways in which fruiting structures are produced and dispersed will say a lot about the ecosystem which they inhabit and can also say a lot about the evolutionary history as well. So examples here such as salmonberry, that bright orange berry, or salal, which are both sought after by wildlife in comparison to something like that fruit of the California uh, buckbread or coffee berry, which not all organisms, all animals will want to be consuming, though certain birds have a real fascination with. And so we can start, and also with that rhamnus species, with that coffee berry, a berry where it improves in its likelihood of germination when it's passed through the gut of an organism. Also, we can think of dispersal mechanisms such as the samaras that appear on those sort of moth-winged uh, seed structures that are maple tree in the upper right-hand corner, which help to flutter the seeds away from the parent tree. Or in looking at this papacy plant, this plant with all of these hairs in the bottom left corner, we can see that there's great opportunity for wind dispersal. Even an extreme over on the far bottom right, where we can see examples of Arceuthobium, which is a mistletoe that grows on conifer trees. And it has these very sticky tissue that allows it to get onto the feet of birds that stand near that tissue. And also, in some cases with Arceuthobium, even explosiveness, so an hydraulic release when heat is applied to that tissue. So in thinking about the time of year that plants flower, how they grow, how they respond to their environment, we are describing something called the phenology. And knowing phenology is really important when thinking and considering conservation of species, or when thinking about and considering the removal of maybe an invasive species. So this is, of course, I think especially important if maybe you're trying to collect seeds, trying to collect those seeds at a time of year when you know that there'll be the greatest likelihood that there won't be maybe pathogens that have arrived on those seeds and may dampen the likelihood that those seeds will germinate, or knowing what sorts of conditions such as freezing or other conditions would be required to allow for those seeds to germinate, which could include maybe fire scarring or acids that would be a throwback to ideas like the stomach acid that we discussed earlier with the frangula, with the coffee berry. And also just knowing if you're working with vegetative reproductive structures, like cuttings or bulbs, when would be the right time of year to harvest those structures with respect to when there would be the most stored energy and the most likelihood for roots and growth to emerge from those harvested structures. And when it comes to these sorts of questions, there's no one answer. It's going to be very dependent on the environment in which you work, the variation in annual and seasonal climate, hill slope even, that will all affect phenology and really getting to know your system. And of course, that also speaks to invasives as well, because we can find that sometimes killing an invasive or pulling an invasive at a certain time of year before, say, seeds have formed doesn't necessarily mean that those seeds won't still come to fruition after that plant has been removed. And we see that with various grass species where they may still be able to cure and produce mature seeds even after killed. So being able to time when you do your work in the natural landscape is very important, and it requires simply having a good foundational knowledge of your system and the organisms you work with. So to wrap things up, I think that for many of us as folks who care about California plants and who think about the California Holistic Province and its diversity, conservation and protection of these species going forward is vitally important. And that goes back to a lot of what we discussed today in terms of management strategies, but also in how we maybe collect seeds and preserve local diversity or maintain species that may be at the edge or facing extinction. That's especially, I think, a problem here in California because of the landscape diversity we've identified in this class, we can see lots of unique habitats where there may be refuge for some species that exists nowhere else, or there may be variability that means that species at different ends of their range may be locally adapted to different factors. So when thinking about conservation of species, whether that's moving a species from one end of its habitat to another to assist in providing genetic diversity that would be needed to meet climate change or other anthropogenic variation, or when thinking about things like assisted migration, I think it's important that we recognize that this is as much a philosophical question as it is ecological, in the sense that we can't always anticipate what the future will bring. And that then returns us to the importance of wanting to think about physical attributes and the physiology of the plants that we study. To close this out, I guess I would say that, we think about conservation oftentimes in a very static sense. Pinnacles National Park, seen here, or Yosemite, these are places that were conserved as national parks. And in so doing, set aside as places that would be conserved, but not in a passive sense. Anything that is worth protecting is going to require time and patience and effort to maintain those spaces in perpetuity. And I think that as we look at the California landscape and we consider the role that humans have really always played in these systems, we have to always put ourselves back in that paradigm and always recognize that the landscapes that we look at, we are as much architects as the rain and the wind and the sunlight and the soil conditions. And so moving forward in how we approach conservation ecology, it's important that we always take into consideration those factors. And just as people 10,000 years ago used fire and other tools to manage the landscape for their needs and to promote regeneration and growth in that landscape, those traditions, fortunately, are returning to the landscape in the form of approaches to fire management that borrow from traditional ecological knowledge and reincorporate those practices. And also we see that tradition carried forward in the work of herbarium specialists, in the work of researchers and conservation biologists, and also in the work of an informed citizenry that can help shape policy and legislation that can further protect the protection of these spaces in the coming centuries. So with that, I think we are at the end of our time here, and I'd like to pass things back over to Amy, and there's some linked resources to ideas for seed storage and best practices with seed collection, and we'll open things up for questions.